Hello everyone. Today we'll be talking about restriction enzymes and digestion with the restriction enzymes, and we'll be also talking about the cloning of the digested fragments. We'll also talk about the entry-level vectors like PGMT easy vectors and other type of different vectors available in the market. In the last video, we made an amplicon of CDS for SWIT10 gene, and in this video we'll be talking about how we can insert a restriction enzyme site at the beginning of the fragment and at the end of the fragment, so that we can exactly clone the CDS sequence only, not like UTI regions or anything. So to clone this one, we have 4 different steps that we need to follow. First, we need to find an entry clone or entry level vector like PGMT easy vector or POC57 vector. And second one is insert that restriction enzyme sites in this fragment. In this CDS fragment, we can insert the restriction enzyme sites. And the third one is the digestion with restriction enzymes and cut the fragment so that we can insert together. And the fourth and the final step is going to be the ligation, and we ligate this fragment with the entry clone or another plasmid so that we can make a plasmid with this CDS sequence. So let's go to Google first, and we can look for PGMT easy vector, PGMT easy. So this is a plasmid sold by Promega. So we're not going to Promega site, because if you want to order the plasmid, you can go to Promega site. But for now, we will go to SnapGene and we'll download the sequence. This is PGMT easy plasmid or vector. So let's download the sequence for SnapGene. So here is download plasmid. We'll just download the plasmid and open it. And after opening the plasmid, what we need to do is we need to identify the restriction sites. So there are different labels you can see. So first we need to do is we need to go to enzymes and look for different enzymes that can cut at one place or 2 places. So here is unique 6 cutter only. So we can do unique and dual cutter, means we'll look for enzyme that cut at one site and also that cuts at 2 different sites. And we'll go to the map and see the enzyme. And in the map, mostly we have to go to the, okay, so this is 80 sweet gene. We'll go to PGMT enzyme and we'll look for unique cutter and dual cutter. Okay, so let's go to sequence, or let's go to map and see. Maybe let's go to the sequence. So this is going to be LAGG alpha site. So here we have that restriction site. So these are the enzymes that can be used for cloning. One thing we can do is we can use a single enzyme or double enzyme. So single enzyme, single cutting site is represented by bold color. So this is a single cutting site. So if you see any color, any enzyme without bold, then that is cutting at 2 different sites. So EcoR1 cuts right here and here. That means we can choose this EcoR1 site, or what we can do is we can use 2 different sites like EcoRV. No, this is not. This is going to be a blunt end. We can see here, this is a blunt end. We need a staggered end or sticky end. So we can use SPE1 and we can use SalI. Okay. So these are going to be our 2 enzymes, SPE1 on 5' and SalI in 3'. So let's remember this one. And let's take our fragment and copy this one. So let's copy the CDS sequence, this one. And I think SnapGene viewer won't let us edit it. So first we'll edit in Word document, then we'll bring it back to SnapGene viewer. So let's open a Word document, or we can open Pages, that's for Mac. So it's this fragment. Now we need to look the sequence for the restriction digestion. So let's look for the sequence in restriction digestion, that is PGMT easy vector. So we were looking for SPE1 ACT AGT. Okay. ACT AGT. So this is going to be our fragment that we are going to use the restriction site on the 5' end, and we need another on the 3' end. So our another 3' end was SPE1, and we selected SAL1. Okay, we'll look for SAL1, that is GTC GAC, GTC GAC, GTC GAC. Here we don't need to worry about the orientation or reverse complement, because those are same from the both side. That is, they are palindromic, CAG CTG, so they are same from the both side. Yeah, so now we can use this fragment for cloning, and let's look for some more enzyme that you want to add for further cloning. So if you want to do multiple cloning, you can add more enzymes, otherwise this is going to work for now. So let's copy this one. So what we can do is, either we can order this fragment from Genomics or any of the gene synthesis company, or we can order this primer with this 3' overhanging site, and we can order it. So let's design the primer also. So I have added one more restriction site in the sequence on both sides. So this is SPE1, the first red one, and the blue one is SAL1, and I have added two different restriction enzyme. They are the same restriction site, so that is SAC2 enzyme. So let's copy this sequence and take it to SnapGen viewer and create a new file. So cloning suite 10. So now what we'll do, we'll annotate this fragment like what we did before, and I'm not going to show you the detail, I'll just show you the annotated file. This is our final fragment after amplification. So don't confuse with the PCR fragment we amplified before and with this fragment. So there is one more step we need to consider, that is the primer. So in the last lecture we added this as a primer. So if we only add this one as a primer, then we won't have any restriction site to cut exactly in the location we want. And this is the forward primer, and this is the reverse primer. So what happens? So if we use only these two primers, then we won't be able to clone it. So what we need to do is, if we are going to synthesize a gene, then we need to use this whole sequence from here to here and give it to a company for synthesis. However, if we are not going to synthesize a gene, but if we are going to amplify in-house or in the lab, then we need to consider one more thing, is the primer. So we need to design the primer like this. So all these restriction site has to be added in the primer we designed before. So let's use this one primer, and we can increase the site. Oh my God, where is that? Okay, let's add a new primer. Feature, primer, add primer. So we'll do top strand, and this is going to be AT suite cloning forward primer suite 10. Okay, add primer to template. So this is the forward primer we need to order. We need to add all those overhang on the 5' side, and same thing we need to do in the 3' side. So this is our original primer that we designed before. However, to include this SAL1 restriction site we want and the SSC2 restriction site we want, we need to design the whole thing as a primer. So feature or primer, add primer, and then this is going to be a bottom strand, and then we do AT suite 10 cloning reverse primer. Okay, now we are ready to clone. So this is going to be our, we copy this one and order the primer. This is going to be our reverse primer, and this is going to be our forward primer. Then after we have this primer, then we are going to amplify the fragment, this fragment we have. Then we can amplify any gene or CDS sequence with the restriction site, and then we'll be able to clone that fragment. Our next step is to cut this PGMT-EZ vector and the fragment we amplified by using the restriction site we developed after restriction enzyme digestion. So what we can see is, this is, I have already copied the sequence. So this fragment is going to be on one side. This is from the plasmid, and let's see what is the other fragment we can see. Okay, I already copied this sequence. So what happens, this is our CDS sequence, and this is after restriction digestion. So we see only 3 of the nucleotide here, because we used SPE1 here and SAL1 here for restriction digestion. And same thing with the plasmid. This is our plasmid on one side, so this one has been digested by SPE1, and on the other side this has been digested by SAL1. So now we can combine them together, because in this stage what happens, like I cannot exactly show it, but when they have the same staggered end and the overhang is the same, then they are going to be ligated together. And when they are ligated together, we use DNA ligase as an enzyme for ligation. I won't be able to show all this in SnapGene viewer because I don't have the full license. So if we have the full license, we can exactly do this one in SnapGene, not in SnapGene viewer, but we can do in SnapGene. So let's open a new blank file and paste this one and create a 3854 base pair plasmid sequence with our gene of interest. So okay, so it will automatically identify the feature needed. So we'll just use the feature it has identified, add 13 features for the new gene, and we can save it. PGMT EZ and switch chain. So we have this switch chain. So if we want to know if it has switch chain or not, then what we need to do is we need to go and copy the CDS sequence and paste it here. Ctrl F, Ctrl V. Next. So this is our CDS, 80 switch chain. So let's add a feature. I'll give a color also. Let's give bright green color. So this is our 80 switch chain CDS. So let's give the direction also. We can give the forward direction. Okay. So now this CDS has methionine to stop codon. So the first should be methionine, and at last it should have stop codon. So it has ATG and last is TAA. So this will stop the transcription. It will start here and it will end here. So all the remaining sequence are plasmid sequence. So in the future, if we need to clone this gene again, then we can use EcoRI and MLU1 enzymes, and there are multiple enzymes. So these are called cloning sites. Then we can clone this gene again and again in the future, and we can use it for different purpose. So now finally we have cloned the gene, and this one is ready to be transferred to a vector where there is a promoter and terminator, and we can express this gene. So next step is to use promoter and terminator sequence in order to express this gene. If not, we have successfully cloned this sweet 10 gene. So this is the 5th lecture. So it took us 5 different lectures to clone a single gene. So in the coming lectures, we can talk about how we can use this gene for different purpose. So if we want, we can play around on this plasmid. So we have only added this 80 sweet 10 CDS region. So what happens? So in the previous plasmid without insertion, there was a ligase gene. So in the absence of that ligase gene, so we have inserted this new CDS sequence in between the ligase gene. that was already present in the plasmid. So we want to see the effect of ligation. So this plasmid has ampicillin resistance. So if we want to really work on this plasmid, then we need ampicillin as a selectable marker. So and other good thing is it has two primers, M13 reverse and M13 forward. So what happens if we use these primers and send for sequencing, then we can exactly sequence the clone fragments. So from the reverse side we can do M13 reverse, and in the forward side we can do M13 forward for sequencing purpose also. Let's look at this CDS fragment, and we can actually translate this one because this is a gene fragment or CDS sequence, and this is going to be translated. So if we express this one in any plant or any organism, then we get this protein sequence. So this is all for this lecture. Thank you so much for watching this video, and if you want to learn more about genomics and bioinformatics, please subscribe to my channel and like and comment my content. Thank you so much, and I'll see you in next video.